In this video, we will discuss about N-Glycosylation alteration in the chronic inflammation. So basically, the chronic inflammation will lead to the innate immune response, and that will cause alterations in the immune cells. And now we will understand this innate immune response in terms of neutrophils, monocytes, and NK cells. So some of the major cytokines that will cause innate immune response are this INF alpha, interleukin 2, IFN 1, and other this IFN gamma. So all these cytokines will cause the innate immune response, and that will leads to this N-glycosylation. So we will start with this neutrophils. So when these neutrophils get interacted with these uh, inflammatory cytokines, it changes that will occurs within the neutrophils leads to the leads to this change in the N-glycosylation in a way that it will cause this MAC1 structure formation along with this N-glycosylation structures. Along with this, the neutrophils that will release other neutrophils in terms of the HNE will also contains this N-glycosylation with this with a sialic acids with some sialic acids. So these sialic acids will contributes towards the innate immune response, and this modified glycan structure, that is MAC1 with this DG sign glycan structures, helps in the activation of the helps in the activation of the dendritic cells. And when these dendritic cells get activated, the pro-inflammatory because of the pro-inflammatory cytokines, it will lead to down regulation of these enzymes such as this ST3-GAL1, ST3-GAL6, that will lead to the decylated glycan structures on the inflammatory on the mature dendritic cells. So these are about the dendritic cells, and also these Siglec formations will occur in the mature dendritic cells because of the innate immune response, and that will cause this decrease in the ROS and NET formations by affecting or by generating immune response against this bacteria, that is PA bacteria. And when the at the same time, these cytokines will also attract these monocytes and other leukocytes such as NK cells to reach to the or recruit to the at the site of infection. And monocytes, when it's come in the context of this TNF alpha and IFN gamma, it will differentiate towards this M1 macrophage, and during this differentiation there is a down regulation of this ST3-GAL1 and up regulation of this FAT1, 2, 3, that will cause the glycan structures that will cause the glycan structure which are devoid of the sialic acids on the M1 macrophages. At the same time, these M1 macrophages will interact, will cause celiac formations on its cell surface. At the same time, it will also release some of the soluble celiac one within the extracellular matrix. And at the same time, this M1 macrophages also helps in the cleavage of this galactin 3, and that will contribute, that work is to contribute towards this chronic inflammation. And this complex, this IFN beta and GAL1 complex act as a feedback loop that get dysregulated during the chronic inflammations. The chronic inflammation will prevent this complex formations. And at the and one more function about this celiac one is that it will interact with this RA synovial fibroblast and prevents the prevents its function, or we can say that it's a immune response by the M1 macrophages against this RA synovial fibroblast, and it can interact with the sialic acids along through this celiac-1 receptors on the cell surface. And there are some cases where this M2 macrophages, which has a pro-tumorigenic response, will also formed when there is a when there is a interactions with interleukin 4 and interleukin 16, that will cause the upregulation of this ST3-GAL1, and this M2 macrophages, because of the upregulation of this ST6 not ST3, ST6 Gal1 will cause the sialic acids formations, and that will prevent itself from interacting with this galactin 1. Thereby, it can does not get affected by the galactins, and it can work on its and pro-tumorigenic process. Now let us understand about this NK cells. So for the NK cells, there is a when it's get interacted with the sialic acids, when it's interacted with the cytokines, because of this FCYR3A present in the NK cells, it will lead to this high structures formations, and that will trigger the ADCC, that is antibody dependent cell cytotoxicity. At the same time, it will also upregulate the enzymes that will glycotransferase that will helps in the silations. Thereby, these NK cells will have more silated glycan structures after the innate immune response. So I hope you got a basic idea how the N-glycosylation alternation occurs in the basic innate immune cells, such as this in case of neutrophils and macrophages. That is M1 macrophage, there is a devoid of silations, and in the case of M2 macrophages and these NK cells, there is upregulations of the silations. If you need to understand more about this and glycosylation alterations in chronic inflammations in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.